Food chemistry is the study of chemical processes and interactions of all biological and non-biological components of foods. The biological substances include such items as meat, poultry, lettuce, beer, and milk as examples. It is similar to biochemistry in its main components such as carbohydrates, lipids, and protein, but it also includes areas such as water, vitamins, minerals, enzymes, food additives, flavors, and colors. This discipline also encompasses how products change under certain food processing techniques and ways either to enhance or to prevent them from happening. An example of enhancing a process would be to encourage fermentation of dairy products with microorganisms that convert lactose to lactic acid. An example of preventing a process would be stopping the browning on the surface of freshly cut red delicious apples using lemon juice or other acidulated water. History of food chemistry. The scientific approach to food and nutrition arose with attention to agricultural chemistry in the works of J. G. Wallerius, Humphrey Davy, and others. For example, Davy published Elements of Agricultural Chemistry in a course of lectures for the Board of Agriculture in the United Kingdom, which would serve as a foundation for the profession worldwide. Going into a fifth edition. Earlier work included that by Carl Wilhelm Scheele, who isolated malic acid from apples in 1785. In 1874, the Society of Public Analysts was formed, with the aim of applying analytical methods to the benefit of the public. Its early experiments were based on bread, milk and wine. It was also out of concern for the quality of the food supply, mainly food adulteration and contamination issues that would first stem from intentional contamination to later with chemical food additives by the 1950s. The development of colleges and universities worldwide, most notably in the United States, would expand food chemistry as well, with research of the dietary substances, most notably the single-grain experiment during 1907–11. Additional research by Harvey W. Wiley at the United States Department of Agriculture during the late 19th century would play a key factor in the creation of the United States Food and Drug Administration in 1906. The American Chemical Society would establish their Agricultural and Food Chemistry Division in 1908, while the Institute of Food Technologists would establish their Food Chemistry Division in 1995. Food chemistry concepts are often drawn from rheology, theories of transport phenomena, physical and chemical thermodynamics, chemical bonds and interaction forces, quantum mechanics and reaction kinetics, biopolymer science, colloidal interactions, nucleation, glass transitions and freezing, disordered or non-crystalline solids, and thus has food physical chemistry as a foundation area. Water in food systems. A major component of food is water, which can encompass anywhere from 50% in meat products to 95% in lettuce, cabbage, and tomato products. It is also an excellent place for bacterial growth and food spoilage if it is not properly processed. One way this is measured in food is by water activity, which is very important in the shelf life of many foods during processing. One of the keys to food preservation in most instances is reduce the amount of water or alter the water's characteristics to enhance shelf life. Such methods include dehydration, freezing, and refrigeration. This field encompasses the physiochemical principles of the reactions and conversions that occur during the manufacture, handling, and storage of foods. Carbohydrates. Comprising 75% of the biological world and 80% of all food intake for human consumption, the most common known human carbohydrate is sucrose. The simplest version of a carbohydrate is a monosaccharide which possesses the properties of carbon, hydrogen, and oxygen in a 1:1 ratio under a general formula of CnH2 non where N is a minimum of 3. Glucose is an example of a monosaccharide, as is fructose. Combine them in the picture shown to the right and you have sucrose, one of the more common sugar products around. A chain of monosaccharides form to make a polysaccharide. Such polysaccharides include pectin, dextrin, agar, and xanthan. Sugar content is commonly measured in degrees Brix. Lipids. The term lipid comprises a diverse range of molecules and to some extent is a catch-all for relatively water-insoluble or non-polar compounds of biological origin, including waxes, fatty acid-derived phospholipids, sphingolipids, glycolipids and terpenoids, such as retinoids and steroids. Some lipids are linear aliphatic molecules, while others have ring structures. Some are aromatic, while others are not. Some are flexible, while others are rigid. Most lipids have some polar character in addition to being largely nonpolar. Generally, the bulk of their structure is nonpolar or hydrophobic, meaning that it does not interact well with polar solvents like water. Another part of their structure is polar or hydrophilic and will tend to associate with polar solvents like water. This makes them amphiphilic molecules. In the case of cholesterol, the polar group is amiro. Lipids in food include the oils of such grains as corn, soybean, from animal fats, and are parts of many foods such as milk, cheese, and meat. They also act as vitamin carriers as well. Food proteins. Proteins compose over 50% of the dry weight of an average living cell and are very complex macromolecules. They also play a fundamental role in the structure and function of cells. Consisting mainly of carbon, nitrogen, hydrogen, oxygen, and some sulfur, they also may contain iron, copper, phosphorus, or zinc. In food, proteins are essential for growth and survival and vary depending upon a person's age and physiology. Protein is commonly obtained from animal sources. Eggs, milk, and meat. Nuts, grains and legumes provide vegetable sources of protein, and protein combining of vegetable sources is used to achieve complete protein nutritional quotas from vegetables. Protein sensitivity as food allergy is detected with the ELISA test. Enzymes. Enzymes are biochemical catalysts used in converting processes from one substance to another. They are also involved in reducing the amount of time and energy required to complete a chemical process. Many aspects of the food industry use catalysts, including baking, brewing, dairy, and fruit juices, to make cheese, beer, and bread. Vitamins. Vitamins are nutrients required in small amounts for essential metabolic reactions in the body. These are broken down in nutrition as either water-soluble or fat-soluble. An adequate supply of vitamins can prevent diseases such as beriberi, anemia, and scurvy, while an overdose of vitamins can produce nausea and vomiting or even death. Minerals. Dietary minerals in foods are large and diverse, with many required to function, while other trace elements can be hazardous if consumed in excessive amounts. Bulk minerals with a reference daily intake of more than 200 mg day are calcium, magnesium, and potassium, while important trace minerals are copper, iron, and zinc. These are found in many foods, but can also be taken in dietary supplements. Color. Food coloring is added to change the color of any food substance. It is mainly for sensory analysis purposes. It can be used to simulate the natural color of a product as perceived by the customer, such as red dye to ketchup, or to add unnatural colors to a product like Kellogg's Fruit Loops. Caramel is a natural food dye. The industrial form, caramel coloring, is the most widely used food coloring and is found in foods from soft drinks to soy sauce, bread, and pickles. Flavors. Flavor in food is important in how food smells and tastes to the consumer, especially in sensory analysis. Some of these products occur naturally like salt and sugar, but flavor chemists develop many of these flavors for food products. Such artificial flavors include methyl salicylate, which creates the wintergreen odor, and lactic acid, which gives milk a tart taste. Food additives. Food additives are substances added to food for preserving flavors, or improving taste or appearance. The processes are as old as adding vinegar for pickling or as an emulsifier for emulsion mixtures like mayonnaise. These are generally listed by E number in the European Union or GRA by the United States Food and Drug Administration. Bibliography. Fennemer, O.R., ed. Food Chemistry, 2nd edition, revised and expanded. New York, Marcel Decker, Inc. Francis, F.J. Harvey W. Wiley. Pioneer in Food Science and Quality, in A Century of Food Science. Chicago, Institute of Food Technologists. pp. 13-14. Potter, N.N., and J.H. Hotchkiss. Food Science, 5th edition. New York, Champman and Hall. pp. 24-68. U.S. Food and Drug Administration. Everything added to food in the United States. Boca Raton, Florida. C.K. Smoley.